morning, everybody. My name is Deepak Srivastava, and I'm delighted to welcome you today to the 2025 Ogawa-Yamanaka Stem Cell Prize event. This is a very special day for the stem cell community worldwide, for Gladstone, and for, I think, scientists all over the world and patients who really care about regenerative medicine and the ability to address some of the diseases that have been so difficult for so many decades to address, that we have great hope that some of the discoveries that have been awarded by this prize over the years will really make a significant difference. This is really one of our flagship events here at Gladstone, and I'd like to welcome not only the folks here in the audience, but also our online audience. We are live streaming this, and I know many folks in Rudolf's laboratory in Boston must be watching, and so many of his trainees all over the globe, and I'll say more about that in a moment. In a moment, Shinya Yamanaka will say a few words about the actual prize, but this has become one of the world's leading stem cell prizes, and Shinya will say more about the award and its origins in a moment. But we are lucky to have a wonderful selection committee that helps us administer this each year. This includes myself and Shinya, but also George Daly, Hideyuki Okano, Lawrence Studer, Fiona Watt, and so this is really a global group, and they spend a lot of time looking through the wonderful nominations that we get. And since 2019, we've been fortunate to partner with Cell Press on this award, and they do a nice write-up on the recipient that'll be in Cell Stem Cell in January. And this award really got started with Hiro and Betty Ogawa, who were very close to Shinya and proposed doing this award. And I'll ask Shinya now to come up and say a few words about that. For those of you who I think everybody in the audience knows, but online, Shinya Yamanaka is one of our Gladstone investigators. He trained here in the 1990s, went back to Kyoto University, where he did his beautiful work on cellular reprogramming. We are fortunate to recruit him to come back to Gladstone and run labs in both places since 2007, which he continues to do. And of course, he received the Nobel Prize in 2012 for his discovery of induced pluripotent stem cells and cellular reprogramming. Shinya continues to run a laboratory here and is director emeritus of CIRA at Kyoto. Shinya?
Good morning, everyone. Yes, so as Deepak mentioned, Hiromitsu Ogawa and Betty Jean Ogawa are my very close friends, and also my wife Chika's mentors. Hiro was a very successful businessman. He was the first Japanese, with the great help of Betty, who made his company public at the New York Stock Exchange market. He was very successful. After his success in business, he decided to give back to society. He helped many organizations, mainly in Bay Area, including like San Francisco Opera and Asian Museum, and also Gladstone Institute, including myself. So I remember it was in April in 2014, 11 years ago. They were in Japan. After enjoying cherry blossoms, Hiro and Betty came to my office in Kyoto, and they proposed to start a stem cell prize. They insisted to have this name, Ogawa-Yamanaka Award. I insisted to have just their name, Ogawa Award. I insisted, but they insisted. So we said, okay, they had to come back to San Francisco And I'll be in San Francisco in like 2 weeks. Let's discuss it again in San Francisco. But soon after, Betty became very sick. And like 2 hours before I landed at SFO, Betty passed away. So to me, this prize is really the last will of Betty. And we were able to start this award in 2015. So far, we have awarded many, many brilliant scientists in our field. This year means a lot to me. As you know, Isaac Newton, he said, if I have seen Father, it is by standing on the shoulders of giants. This year's recipient is my giant. You will hear a lot more about Rudolf Jaenisch, but he is really my giant. Without his pioneering work in this field, we could have never generated iPS cells. So many, many congratulations, Rudolf. Thank you very much.
Thank you, Shinya. It's really true, and the whole field recognizes that, and that's why it's such a delight. Our committee, when we met, and Dr. Jaenisch's name came up, it was immediate that everybody said, yes, of course, we must award this. So Rudolf Jaenisch, this year's recipient, is receiving the prize because of his pioneering contributions in epigenetics and stem cell biology, which have really advanced our understanding of gene regulation, cellular reprogramming, and the potential of regenerative medicine, as I mentioned earlier. And by way of background, Rudolf did his MD in Munich, and then did his postdoctoral fellowship with Arnold Levine at Princeton. And it was during this time that he began working on tumor viruses and studying gene expression. And really you can track people's career along the way. Even the origins that may seem unrelated are in fact related often to their discoveries later on. And it was the study of tumor viruses and their regulation of gene expression that led him to study the epigenetics of gene regulation. And it was his work on viruses that led him to insert for the first time external DNA into an early embryo and create the first transgenic mice that we all utilize frequently. That is something that evolved out of Dr. Jaenisch's work. And then ultimately using that same kind of study of gene expression and gene regulation to understand developmental biology, cell fate decisions, and ultimately cellular reprogramming. After doing his work at Princeton and then also a period at the Salk Institute, Rudolf went back to Germany and was for 7 years at the University of Hamburg, and then was recruited to come back to the United States as a founding member of the Whitehead Institute as it was starting in 1984, and also as a professor at MIT. And he's been there ever since and has had just a tremendous impact on our field scientifically with his own work in his lab, but maybe just as important or even more impactful through his trainees, which are just a remarkable group of people who emerged as leaders all across the world in so many areas, many of whom are right here in the Bay Area and in this room, and I know are watching online as well. And of course, his contributions have been recognized with many awards. He's been elected to the National Academy of Sciences, National Academy of Medicine, American Academy of Arts and Sciences. He's received the National Medal of Science, the Wolf Prize, and the March of Dimes Prize in Developmental Biology, among many others. He's also served as a former president of the International Society for Stem Cell Research and has been a great leader in our stem cell community for many, many decades. Hiro and Betty Ogawa, when recognizing our award recipients every year, had made a point that they wanted to always recognize the spouses, male and female, that would have been part of the team that really it takes to make the personal and professional sacrifices to achieve at a very high level. And that is always the case. And so I'm delighted that Rudolf's wife, Alexa, who's a physician in her own right, is here today, as well as their son, Johan, who's here and is in the biotech world, in plant biotech, working with the company here at Berkeley, although living in Boston. But it's wonderful to have you all here today for this very special event. And at this time, I'd like to ask Rudolf to come up to the stage and accept his award, which is a trophy. And let me say a word about this trophy for a moment, which is one that we designed specifically for this award, that the top of it represents the epigenetic landscape, the Waddington landscape, which is particularly fitting for cellular reprogramming. And today's award, it's a local San Francisco artist who made this, and every recipient thus far has received the same award. And along with that beautiful trophy is a $150,000 check that comes with this. So Rudolf and Ksenia, please join me. Congratulations. It's very, and Ksenia, please join. The stage is yours.
Well, thank you very much, Deepak. That was such a nice introduction. I must say, when I got the call from Deepak informing me that the committee had chosen me, I was in a way shocked. I was very surprised because I thought it would be not possible that I would be honored this late in my career with this prize. So from that point of view, I was very, very grateful, very honored, and I think that's it's an incredible honor for me. So I want to give you briefly when I talk about really gene editing disease, transgenic mind stem cells, because I was involved in this. And doing the outline of my talk, so one of the crucial technologies in biology has been the genetic manipulation of cells and animals. And my talk will provide a brief history of some of the, what I think, important steps in developing this technology, and mention my modest involvement in some of those involvement in events. I've frequently changed the emphasis of my research, and I will point out the different focus for each of the past decades, so by decade. So what I think was the most important events. So in the 1970s, I was involved with transgenic mice, with SV40, and with retroviruses. And I joined as a postdoc, Princeton University, Arne Levine's laboratory, as Deepak mentioned, and I worked on SV40 DNA replication. So SV40 is a tumor virus, and when you inject it into mice, it makes sarcomas. So I naively thought, why only sarcomas? Why not brain tumors or liver tumors? It was a very naive, simple question of tropism. And I wasn't, maybe could infect those cells or could infect these cells like brain cells, but could not transform them. And then I read this paper by Beatrix Mintz. Beatrix Mintz was one of the eminent developmental geneticists. And in this scheme, she describes how she make what she called allophenic, we call it chimeric mice, by aggregating a black and a white embryo and getting the striped mice. And from the pattern of the stripes, she tried to conclude something about how pigment system was set up. So it was a retrospective analysis, very difficult to understand. And I didn't understand a word of this when I read this. But what I found very interesting was that combining or having embryos culturing in a petri dish and making a mouse that blew me away. And I thought, using this technology, you could possibly answer the question I had asked, because I could introduce a virus into this early, into the early, it doesn't work, into the early embryos, it would end up in the liver and the brain. So I proposed this experiment to her, and she was very kind and interested, but she was very skeptical. Who was I? I was a postdoc from molecular biologist. And then she agreed, and she told me everything I know about embryos. And we really managed to make these mice which carried SE40 sequences. The problem with these mice was they didn't get tumors, because as we now know, they were silenced. And there was no germline transmission, because I didn't know, I didn't realize at this point how rare this is and how in chimeric mice. So it was only later, when I got my first job at the Salk Institute, that I generated some germline transmission of Maloney virus. So the 80s, it was a decade of transgenics, and the main players were Ralph Princeton and Palmeter, and many others. And it was really the decade where they expressed genes ectopically in mice. The famous experiment was the growth hormone experiment, but oncogenes, and many oncogenes were expressed, but I don't think they were very informative, these experiments, because they were informative for diseases. Also, mutant mice were isolated by insertional mutagenesis, and the viruses were used as probes. And then I moved to Hamburg, and we generated an insertional mutation in the collagen 1 gene. And so the lessons from infecting embryos with virus was exposure of early embryos to virus allowed germline integration, was very clear. Virus expression was controlled by the epigenetic state, the DNA methylation, as it turned out, was very important. And mice carrying SE40 sequence developed no tumors because they were silenced. And then we could get insertional mutagenesis. So the 90s was a decade of cell technology. So we could make any mutation now. And we made beta-2M was the first mutation made in mice, DNMD1, MyD, MYF5, PDNF exists, and so on and so forth. And it was really embryonic development methylation and epigenetic regulation of imprinting, X inactivation, and cancer. And then I moved to the Whitehead Institute. Baltimore, David Baltimore was a founding director. And this mutation, the inactivation of DNMT1, was a very informative mutation. It informed us about imprinting, the X inactivation, and cancer. So really, it was a very useful mutation. In the 90s also, was when nuclear transfer was invented. When nuclear dolly was made, and therapeutic and cloning and therapeutic cloning was described. And there's Dolly. And it was Ian Wilmut who generated Dolly, and then a year later, Yanagamachi's laboratory from Hawaii showed it worked in mice. When I read this paper, I was very excited. It was really what he wanted to do. And so, so I immediately arranged for collaboration with them, and a few years later, we showed this can be used for correcting a defect. The 2000s were the decade of reprogramming and IPS technology for medical applications. And if you think about mice, it was shown it works, but in human, this was for many reasons not an option for many I'm going to go into. So it was really clear to find out how does the egg accomplish this. And as you all know, that was Shinya's contribution. And the paper, the key paper which came out here, I was at the meeting when Shinya presented this first, and many people were disbelieving. can't be that simple. I believed it immediately because I knew Xenia and everything. So anyway, but the mice he made, or the cells he made, were only partially reprogrammed. They couldn't make chimeras, and they were partial. But it showed the principle was there. And then a year later, really 3 labs, Xenia's lab, my lab, and Konrad Redinger's laboratory showed this technology did accomplished all this, and it really started the explosion of the field. And the lessons from all this was reprogramming pluripotent stem cells could be done without nuclear transfer, and the medical applications of iPS technology was demonstrated by correcting a couple of diseases. 2010s. So I should say that homologous recombination was really able to generate mutant and models, but it worked only when you had ADS cells. So it only worked in mice and humans because they were the only species where stable ES cells were, because you had to select for the rare event. But the CRISPR technology, you didn't have to do this. It was just so efficient. So it really changed the field enormously. And we realized this one-step generation of mice carrying mutation. So really, one could 8 mutant alleles at one. We could mutate in ES cells. We could do it in embryos, and one could even do point mutations in embryos. So it was really a technology which was used widely, and it was it changed the field. And also epigenetic editing was important, and it was pioneered by Jonathan Weissman, David Liu, and many others. And we used this by really to reactivate the methylated FMR1 gene in Fragile X syndrome and to reactivate the silent MECP allele in Rett syndrome. So I was interested in autism, and most autism cases are sporadic without clear genetic evidence, but there's some which have a clear genetic basis. And I'm going to concentrate on Fragile X and Rett syndrome, which is very high incidence. And so Fragile X, give me just a brief summary of this. Fragile X, the FMRs on the X chromosome, it's caused by silencing of the FMR1 gene, and it's due to an expansion of a repeat above 200, and that gets methylated and silenced. So we targeted the guide RNA and TET to demethylate it, and so that we targeted the repeat. And this shows indeed there was a targeting what worked. The guide RNA got removed methylation, and when you looked at the methylation of the CpG islands where the promoter is, it was, we targeted the repeat, but it demethylated also the promoter. So really this worked well. The demethylation of the CpG repeats demethylated the upstream and not targeted FMR1 promoter and restored normal FMR expression. Rett is a mutation of the MECP2 gene. It's X-linked, onset of 1 year of age, and this very severe mental retardation, stereotypic hand movement, and so on and so forth. And the challenges for Rett were, the original model from Adrian Bird was, MECP binds to methylated DNA and acts as a repressor. However, the normal function of MECP1 gene expression is very complex. And the proposed functions were, it acts as a repressor and it acts as an activator, just opposite for many genes. So we looked at this, and it turned out that MECP2 recruits POL2 on many, many genes, and really is important for the activity of the gene. So it acts as an activator of gene expression. And the model is that in normal neurons, MECP2 recruits POL2, and that leads to expression of thousands of genes. But in Rett neurons, either because MECP2 cannot bind anymore, or the interaction between POL2 and MECP2 is disturbed by a mutation, it leads to dysfunction, to non-expression of many genes. So it's a very different picture of how MECP2 works. Now, the last decade, the 2020s, the pandemic, I switched the focus of my lab to coronavirus biology, which just combines my background in retrovirus biology and ES cell and manipulation technologies. And it really started on a kayak trip in 2020 in Maine with Rick Young, a colleague and friend. And we sat there and talked about what else? Corona. And because it was under quarantine. And the question was, really, when this is the infection, then early in the infection, you get high titer virus. The virus disappears after 10 days or so, but you remain PCR positive for months, for a long time. Why? And so the hypothesis we came up with, could SARS-CoV-2 RNA be reverse transcribed and retro-integrate into the human genome? Could this explain the long-term PCR positivity? And just contrasting retrovirus, where the virus integrates as an obligatory step into the genome, which can be seen by the fusion of the viral with the cellular sequences. Coronavirus instead replicates it exclusively in cytoplasm, with on the 3' end, the subgenomic RNAs encoding the structural proteins. So we showed that SARS-CoV-2 sequence can indeed integrate into the genome by a line 1 mediated retrotransposition. Now, the footprints of line 1 mediated retrotransposition are a target site duplication and a presence of poly-A site. And the methods to detect it, the nanopore, the advantage is both flanks are detected. The disadvantage is sequencing deaths of 20 times, which normally is done, really interrogates 10 cells. So it's not very sensitive. TAC-MAP, target enrichment segmentation, is another method. It allows analysis of about 20,000 cells, much more sensitive. So disadvantage is you only detect the 3' flank. So in this rate of transposition has been shown by using reporter assays. In cis, line 1 itself integrates with this frequency, but in transrate transposition of marked ALUs or marked mRNA is 300 to 3,000-fold less. So, as I said, we found that SARS-CoV-2 can integrate into the genome of infected cells by Landman-mediated retrotransposition mechanism, and the integration is rare. It's 1 in 1,000 to 1 in 10,000 cells, but also occurs in patients, as shown by looking at published RNA data. And the hypothesis was the integration might contribute to the disease phenotype by eliciting an immune reaction against fused cellular viral antigens. So this was published in 2021, but this publication really was heavily criticized. We got to really, it was difficult to prove the rare L1-mediated genomic integration of SARS-CoV-2 sequences. The major challenge was virus infection is very toxic, the cells die, and you only can really, by sequencing, find C integration. So we developed an assay to isolate cells carrying integrins, because that would give us the opportunity to analyze the mechanism. And the new assay was not using DNA, which the classical reporter assay was, but using RNA directly. And so the RNA, this again shows you the viral genome, the subgenomic RNA on the 3' end, and we made this reporter nucleocapsid or the spike, the blastocidin promoter in the opposite direction on the 3' end, and after, so this cannot be expressed after retrotransposition. Then blastocidin can be transcribed from the 3' promoter. So this avoids any toxicity. It would allow extensive characterization of integrants, and it could be a general assay for genomic integration of RNA. So we made a couple of reporter constructs, the nucleocapsid, GFP, globin, and others, and either plus minus the 3' UTR of SARS-CoV-2 in those. And this shows you some of the data. So it gives you the plasticide-resistant colonies, and if you don't activate the L1 by doxycycline, these cells, you find there's integration, but without doxycycline, there's none in trans. And if you give doxycycline, all of those integrate, and they integrate if the presence of the in the presence of the 3' UDR, what's 3 to 5-fold higher. And there are two clones. One is more efficient than the other. So the conclusion from this is, in cis retrotransposition is most efficient in trans retrotransposition of nucleic capsid, spike, GFP, and globin only when L1 is overexpressed, and SARS-CoV-2 UTR enhances retrotransposition. So in all of those integrants were analyzed by sequencing, and as I said, there's no retrotransposition without L1 overexpression. Many integrants contain full-length sequences of the in vitro transcribed fragment, have a target site duplication, endonuclease consensus motif, polyase sequence, and in this case, it was located on chromosome 12. So RNA transfection is a sensitive assay to detect retrotransposition of any RNA. So the question, including vaccine RNA. Now, of course, this is a very sensitive issue, particularly in this time, and because of obvious political issues. And can a spike RNA, when injected into people, integrate into the genome, is something which is really in the debate. And it's a very sensitive debate. And certainly this possibility is supported by our data on integration of BNT vaccine RNA, and I don't talk about that. So there are rare vaccine complications, very rare. And the post-acute vaccination syndrome is very severe. It can be crippling. When the hypothesis is it's molecular mimicry, antiviral immune response, cross-reacting to self-antigens, Guillain-Barré is one example, or a reaction against adjuvant or stabilizer and others. So there's a paper which came out this year from Iwasaki's laboratory, and the key finding here is that spike protein is detected in serum up to 2 years after single vaccination. So single vaccination was injected, there was no evidence for virus infection, and these patients make the vaccine, not the antibody, but the spike protein in the serum 2 years after single vaccination. What could be the mechanism of this? So that's not clear, but it could be integration. So that raises questions. So if I summarize our data so far, SARS-CoV-2 sequence can integrate into the genome by line 1 mediated retrotransposition, there's no doubt about. The RNA transfection assay is much more sensitive than the classical DNA-mediated assay. It allows analysis of molecular mechanisms. You're looking directly at what the RNA does, and you don't have to add other parameters you have to clarify. And this assay can probably detect the retrotransposition of any cellular RNA, and RNA may integrate in a much more frequent manner than we think. And it's a really big question is a vaccine RNA. That will be a very difficult question to get into. So I want to close with just giving you another experimental system, namely bats. Now bats, as you know, they are thought to be the source for many of these very pathogenic viruses, which in humans like COVID-19. And it's difficult to work with bats. There are about 1,400 species. They're long-lived. They're unique by metabolism, a very high heartbeat rate. And they're a reservoir for tolerant to many of these highly pathogenic viruses like SARS-CoV-2, Ebola, rabies, Marburg, Lyssa, and so on and so forth. And they're full of endogenous viruses like retrovirus, Borna virus, filovirus, parvovirus. And the bats are tolerant to viruses that are highly pathogenic in humans. Why? So can SARS-CoV-2 be studied in the BART system? And so the cell source for BART IBS cells, which were really created by Thomas Zwacker, and they're very IBS by any means, and we found that indeed the mode of infection of BART fibroblasts with SARS-CoV-2. And I give you just a brief few of these data. So if you don't express AC2, you can't infect BART cells. So they need a receptor. But if you do, BART, either immortalized fibroblasts or primary fibroblasts can be infected, as seen by nucleic capsid staining, and as IBAR90, which are human fibroblasts. And they all express, the human fibroblasts, the BART fibroblasts express RNA and protein, but if you look at virus production, the human fibroblasts produce virus, but the BART fibroblasts do not. They express viral gene products, but they don't express, they don't make virus. So somatic BART cells can be infected with SARS-CoV-2, but do not produce virus. And we looked at viral transcripts, viral proteins, by various ways for progeny virus. And I'll give you one example. So human ES cells, BART IBS cells, can produce virus, but human fibroblasts, BART fibroblasts, is a big difference. They cannot. And that you see is the SAS on Vero cells. And when we look by electron microscopy for DMVs, double membrane virus particles, which are thought to be the place where virus is made, then human fibroblasts can make these. Human BART IPS cells can make those, but BART fibroblasts cannot. So they're really effective in generating vectors for virus. So this summarizes those data, where if you compare human ES cells with BART IPS cells, both can make virus, the BART cells less than the human cells. But when you compare fibroblasts, there's a basic difference. Human fibroblasts are able to produce virus, but BART fibroblasts are not. It's a mode of infection. And it's likely to be constitutive expression of type 1 interferon and other innate immune response genes, which are constitutive in BARTs, but inducible in humans. So I think BART cells are ready to fight virus infection much earlier than human cells. And when you look at BART as virus reservoir, then BART says the viral tolerance is really by mutation of STING, inflammasome, furin, and other genes is diminished. But the antiviral host defense by interferon is really, for example, is increased. And we looked at this in my last experiment. When it muffs the mitochondrial antiviral signal, which integrates RIG1 and MDA4, detectors of double-stranded viral RNA, with the downstream genes, when we introduced the BART version of MUFs into human cells, then you can see that the human cells carrying BART mitochondrial ABS, they're really reduced in 3 different cell lines. They reduced the viral titer substantially. It's not the expression if you introduce human MAF into these cells. That doesn't make a difference. So this MAF inhibits SARS-CoV-2 replication in human cells. So if I summarize those BART IBS cells in the coronavirus, the BART IBS cells expressing the AC2 receptor, which is what can be efficiently infected with SARS-CoV-2. The IBS cells produce virus, the somatic cells do not. blood fiber rust, they undergo an abortive infection, and mitochondria seem to evolve the tolerance. So coming back to the beginning of my talk, so the interest in my lab has shifted quite substantially. And it is repeating. In the 1970s, 1980s, we were concerned with transgenic mice, with retrovirus insertion mutagenesis. In the 1900s, ES technology was important, mutations, homologous recombination, better to MD and MT1, and so on and so forth. And were interested in epigenetic regulation of imprinting, exon activation, cancer, and nuclear transplantation. Dolly was really made in this decade. In the 2000s, for the reprogramming IPS technology and medical applications, 2010 CRISPR technology really revolutionized biology. In the 2020, really totally shift was the coronavirus, who combined my background. So I want to finish here and really thank again for the enormous honor to get this award. And I hope you have some questions I can answer. And this is the people who did much of this. The SARS-CoV-2 was Qi Liu, Liu Zhang, Lai Gao, a lot of Chinese, Punan Bishtet, Alexei Richards, and then worked together with Rick Young and Thomas Zwacker, and Frederick X, Sean Liu, and Xin Tang, two former postdocs who have their own laboratories now. Thank you.
Thank you very much, Rudolf. We have time for some questions from the audience.
Well, thank you. This was a wonderful journey. I have a question regarding the integration, which is, of course, very provocative and has, gained some sustained interest because of all the long COVID and persistent viremia, not RNAemia that we see in some of these people. I wondered whether you have tested your in vitro transcription assay also with other coronavirus RNAs, and whether that's SARS-CoV-2 specific or...
We have not the coronavirus. We have looked for other RNA virus, positive strand, like influenza and Zika, and we haven't found it, but that was really looking at published data. Yeah. So I think these are negative data which are not very strong. So I don't know. I would be surprised if it's really unique for corona, but that's what we really the only system we looked at.
And my other question is if you see a correlation between, virus RNA replication strength. So if you have very high levels of RNA, if you have a very vigorous infection ongoing, especially RNA replication, do you think that determines whether the rare events could, happen, more frequently, and whether, other lowly infected or potentially, cells exposed to the vaccine mRNA might not, have a chance to undergo this rare event?
We know only when you have more RNA, by transfection also, you get more integrations.
Yes.
That's a clear correlation. But by various ways we could do this. But otherwise we don't know.
Alex?
I also want to ask the same topic, but from the opposite perspective. Do you think you could learn the rules about if it's a 3' UTR sequence or some other sequence determinant, if you wanted to increase the genomic integration of RNA?
So the UTR, we looked at this. So when you look at the UT3 from COVID, from SARS-CoV-2, this has 2 stem cell loops. We deleted these loops. And if the first loop is deleted, that really takes away the stimulant of 3 to 5-fold integration. The other one doesn't have that. So it looks like there's a stem cell, stem type of structure, which is important, which has been seen in allo sequences too, also. So we believe that is one of the issues that provides integration enhancement for all RNAs we tested. But we don't know what the mechanism is.
Maurice?
Beautiful talk. I was wondering whether you also looked in the bad cells, whether you also see integration in the bad cells.
Yeah, we did. So the bad cells are very weird. So they have a lot of endogenous retroviruses, and they have actually retroviral DNA in the cytoplasm, which is pretty amazing. Normally it's toxic. When we infect them, yes, they make lots of DNA copies, and 10% of those integrate. So there's integration occurring. We have done this. And what's amazing is the BART genome is very small compared with the human genome. And when they integrate, why doesn't it grow? So that's very strange. So something we don't understand quite. So the BART must have some way to integrate, but then they restrict the amplification, the increase of the size of the genome, and we don't understand what the mechanism is. So the BART cell biology is quite enigmatic.
We have a question from online, and I think the question is, what has Catalin Greco said about the vaccine integration? Yeah.
So, vaccine integration. So I'm a little worried about this, I must say, because I mean we were so heavily criticized, and now if you need very strong data to do this, so I'm aware of this. But I don't want to be on the wrong side of the debate, which I would be, because it would be used by the anti-vaccine lobby as an argument against vaccines, and I don't think it is. But it will be very difficult to control. But on the other hand, this may be a mechanism of genome manipulation or change, which is underappreciated. So I believe it's important to clarify this. So we're facing a dilemma here.
Yeah, I see that. Well, Rudolf, let me ask you a question then. You beautifully traced out some of the milestones over several decades. I have two related questions. First is, what do you think is the next thing that you would put on that slide if you're looking to the future? And I ask that because you've clearly, in each of those, you've been pioneers in those, and you've seen the future before others in many cases.
Well, I mean, the very, very immediate experiments will be to make this is really working for therapy, and I think there's some progress, although it's slower than we thought initially, but Shinya tells me that it's clearly there, and but this is just a very clear extension of what we know. What are the main new questions you're asking? So I think the key questions are, how does it work? How does the brain work? How does this work? And I think that will be very complex questions. So don't ask me how you would do. So this will be something which, but I think these are the main questions which we'll be facing with.
And yes, question here. And I'll come back to my second related question.
Yeah, thank you so much for an amazing talk. In your kind of ending slides, you slightly mentioned that the mitochondria might be related with the tolerance. I think iPSC, they use less mitochondria, they're more heavily reliant on glycolytic. And do you think that metabolic shift kind of affect these tolerance? Do you think for example, brain or heart, they use more mitochondria? So do you think those cell-specific thing and also the cell kind of differentiation stage, metabolic shift, also affect these kind of tolerance?
Yeah, I think these are very important mechanistic questions. We're studying those. So I don't have a clear answer on this, but it's very clear that from the MAF, the BART mitochondria somehow prevent virus replication, or make it abortive, I should say. So that's what we know, but we would like to understand the mechanism more carefully.
So, yes, Benoit?
thank you for, walking us through, what it is really an amazing career. A bit of a provocative question. Is there a favorite of yours in all those different steps in those years?
So my favorite was seeing that Bologna virus can integrate and give rise to that was amazing to me. Seeing through the microscope, looking at the XT plug assay we used, and then seeing that was probably the most amazing finding in my career.
Your virus comments clearly sparked more interest in maybe there were stem cell comments. We have another question on on vaccines, and that is related to your thoughts about broad vaccine development for developing vaccines that have broad spectrum coverage.
So vaccines, the RNA vaccine is such a successful way to do this and will be used for many, many different applications. So we have to be aware of this. This could be, in probably rare cases, could lead to adverse effects. And I think the Iwasaki paper from the after single vaccine integration, getting spike protein made in the serum 2 years after vaccination without evidence for virus infection we want to explain that. So I think it's somewhat worrisome, but it's very rare. So it's not an argument against vaccination. But then, on the other hand, vaccinations, RNA vaccination, will be used for so many different applications. So I think it would be important to know this can happen, and how can you prevent it? Obviously, you can prevent it by inhibiting, we have shown this, inhibiting the reverse transcriptase by an HIV drug. That works. So I think it's important to understand this.
Maybe I'll end with the last question, that is more of a approach and philosophical question that I'm asking actually for the trainees. And that is that, if you look back on your career as you did, you clearly had a direction and a thought process. How might you crystallize your approach to science for younger folks that led to your extraordinary success in this repeated breakthroughs? It's not by chance. And so I imagine you've thought that there is an approach to this.
So I think I got interested in some questions. I was willing to take the risk. So I think if my first experiment with Beatrix Mintz wouldn't have worked, I probably would be a country doctor. so it would have been the risk taken. I took the risk. So I think it's fun to ask the question and then try to understand and try to resolve it by whatever means they are. And that, for me, was the most satisfying and rewarding experience. And not being afraid that it doesn't work. Things don't work in the beginning anyway.
So courage and risk-taking,
yeah.
I think, is probably the theme repeatedly. Well, I want to, before we close, I want to also mention that Andrew and Marcus Ogawa, Hiro and Betty Ogawa's sons, have decided to continue this prize and are going to continue. Their parents set up this prize and made a gift years ago, and their sons, in their honor, will be continuing. So I'm grateful for that. The Roddenberry Stem Cell Center, I'm grateful for. They continue to support our stem cell work here at the Gladstone Institutes. And these sorts of events take a lot of effort, and I want to acknowledge Irma Hernandez and Bethany Taylor, who did the bulk of the work for making today possible. So thank you all for doing all that. And finally, please join me in congratulating and thanking Dr. Jaenisch for coming today and receiving this wonderful prize.